Hello, my name is Hazel, and my presentation is on the general increased CO2 production in yeast using corn syrup in the presence of amylase. So, a little bit background: fermentation is a metabolic pathway that occurs after glycolysis when there's no oxygen present. It's fueled by the regeneration of NAD plus to produce ATP. As you can see in this diagram here, glucose undergoes glycolysis, which produces the 2-ATP, and that creates the pyruvate. However, because there's no oxygen present, the pyruvate cannot undergo the processes necessary for cellular respiration. So then what happens is that the pyruvate will turn into the acetaldehyde, and that will remove two carbon dioxides from those two pyruvates. And then as the acetaldehyde is being converted to ethanol, that fuels the NAD plus or regenerates the NAD plus, which is then needed for glycolysis, which then makes the ATP or powers the ATP creation. So it's one big old cycle, one big old circle. And so this removed CO2 right here is what my experiment is measuring. So my main objective out of these three was, does the presence of an enzyme affect the rate of CO2 production in a yeast cell? So the relevance of this can be seen through ethanol production at the end of fermentation. This ethanol is used as a biofuel. It's a much greener alternative compared to that of crude oil. Additionally, the CO2 byproduct is used in different scenarios like production of beer, bread, and carbonated beverages, the holy trinity. So my materials here, as you can see, corn syrup solution, I had some water, a calcium chloride buffer, yeast cells, Durham tubes, although this Durham tube is smaller than the one we used in class, and a pipette to transfer the solutions. So the tubes with different amounts of substrate and buffer were filled based on my experimental plan on the next slide. They were then placed in a hot water bath. Yeast was added. They were mixed, added to a Durham tube, which was inverted back into their original test tubes, and then they were returned to the hot water bath. And so their volumes of remaining solution were collected and measured every 5 minutes. So, and we would compare that to the original amount of solution that was present at t equals 0. And so the difference between the t equals 0 versus the amount of solution that was present at like 5 minutes, for example, find the difference between that, and that was how much CO2 was present at 5 minutes. So these are my different groups. My negative control groups are called that because they don't have corn syrup in their substrate, or there's no substrate within it. And then my experimental groups here, as you can see, has an enzyme, while the other two groups don't have the enzyme. So, as you can see here with my results, two things to note here that are important: my absolute control and my negative control both fulfilled my expectations. There's no corn syrup present, so it's not going to undergo fermentation. There's nothing to eat. But what did break my expectations was this group 2. As you can see, my group 2 has a larger volume of CO2 produced compared to my experimental group. My group 2 didn't have the enzyme. So when I saw this and I calculated this, I was very confused. And when I confirmed my results with the linear rates of CO2 production, I found that group 2 had the highest amount of CO2 production compared to my experimental group. Although my experimental group does or has a higher production rate compared to that of group 1 and my controls, group 2 for some reason stood out and had the highest rate. So, just for like some discussion here, in order for corn syrup to be processed into ethanol eventually, it has to be broken up into its glucose monomers. Now, yeast cells have their own processes and enzymes that break down the the corn syrup into its glucose monomers, and then it can be brought across the plasma membrane. However, my thought was that if you add additional amylase, then that additional enzyme will break down that glucose already, so the yeast cell does not need to, or it will, lower the activation energy necessary to break those bonds, and so the yeast cell will then not expel as much energy. And as a result, the CO2 production rate would be quicker because less energy would be needed to hydrolyze the corn syrup because it's already had its activation energy lowered by the amylase. So this slide, I kind of just explained that a bit more fluently. And then as those monomers are brought across the cell membrane by membrane proteins, they'll undergo glycolysis and fermentation. So as you can see, this didn't really match my results, as my experimental group with the enzyme present had a lower CO2 production rate than my group 2, which had the highest rate despite having no enzyme. My experimental group had a higher rate of CO2 production greater than every other group except for group 2. So this indicates that there may have been some human error when preparing the solution for group 2, as every other result was consistent. So in terms of comparing it with published results, I picked this paper because of its substrate choice and as well as its similar-esque enzymes. This study partially matched my results, but the paper fully matched my expectations in terms of the role of the enzyme before I did my experiment. The enzyme should have played a role in making the hydrolysis of the cornstarch into its glucose monomers quicker. So this discrepancy, again, from my expectations and the results, is most likely due to human error. This paper I picked because of its focus on CO2 concentration and CO2 production results. This paper did confirm my expectations to specifically the groups that didn't have an enzyme present. Yeast cells have a log period where the production of CO2 is at its maximum rate. And I expected to see this in the groups without the enzyme. So just like a recap, I found that when I have in my experimental group, its linear rate is less than the linear rate of my group 2. However, the experimental group had a higher linear rate compared to literally every other group except group 2. So if I were to do further testing, I would need to, readdress what went wrong with my group 2. So some big takeaways. There is an effect on the rate of CO2 production. It results in a quicker rate of CO2 production when an enzyme is present. I also found that consistent incubation times for every group being tested are very necessary simply because enzyme is such a time valuable solution. So for future study, I would just redo the experiment entirely, and just to make sure that I was doing everything correctly. Additionally, I'd also, test other environmental factors like the acidity, for example.